All right, let's get right into it and deconstruct question 3 from the 2023 AQA GCSE Biology paper. We're going to go through this thing piece by piece, look at what the official mark scheme says, and figure out exactly how to craft those answers that get you every single mark. So here's our game plan. We'll tackle the question just as it comes, starting with the quick 1-mark hits and building up to the longer explanations. For every part, I'm going to show you exactly what the mark scheme is looking for. The whole theme here is communicable diseases, and Salmonella is our main example. This question is designed to test your understanding of the pathogen itself, how our bodies fight back, and how we treat it. So let's dive into the first part. Okay, first up, a classic 1-marker. For these, you've got to be incredibly direct. No long, flowery sentences, no waffle. You just need to state the one key biological fact they want to see. The question is asking for the mechanism. It's not asking how you catch Salmonella. It wants to know what the bacteria are actually doing inside your body to make you sick. And there it is, straight from the mark scheme. For that 1 mark, all you have to write is that the bacteria release or produce toxins. It's interesting, the mark scheme also says they'll accept the word poisons. That's it. 1 simple fact, 1 mark. All right, now for a bigger one, worth 4 marks. This is what's called a leveled response question. That just means it's not enough to just list facts. The logical flow and the structure of your explanation are actually part of the grade. Now you've got to pay close attention here. This is a classic 2-part question hiding in plain sight. To get those top marks, you can't just talk about the animal's immune system. You have to connect that to the second part, how that process stops humans from getting sick. So, a top-level answer needs a really clear logical flow. Think of it like you're telling a story in 3 parts. First, you talk about the initial vaccination. Then you describe what happens if the animal gets infected for real, that secondary response. And finally, you have to make that all-important link to how it protects us humans. This slide basically shows you the examiner's checklist, what they call indicative content. Notice those key words: white blood cells, specific antibodies, and that rapid secondary response. But you know, the real killer point, the one that pushes you into the top marks, is that last one, linking the animal's immunity to humans being protected because they're eating fewer bacteria. Okay, time to switch gears. The next batch of questions is all about a practical investigation, basically testing how well different antibiotics knock out Salmonella. So this diagram shows you the whole setup. That gray area is the agar jelly, which is covered in a lawn of Salmonella bacteria. Then you've got these little paper discs, A through E, and each one is soaked in a different antibiotic. And see those clear circles around some of them? That's the zone of inhibition, which is just the area where the antibiotic has killed off the bacteria. The first question here is all about good lab procedure. It's asking for 2 aseptic techniques. In simple terms, that just means what steps should you take to stop your experiment from getting contaminated by other random microbes that could mess up your results. And the good news is, the mark scheme gives you a whole menu of possible answers. You could talk about sterilizing your equipment before you start, like the Petri dish, or flaming the inoculating loop. You could mention working near a Bunsen flame, since the updraft helps keep airborne microbes away. Even simple stuff like tilting the lid to minimize exposure or taping it shut at the end counts. You just need to pick any 2 of those. Oh, this is a great question because it's about safety, and examiners absolutely love to test safety. Why are the rules different for a professional lab versus a school lab? Well, there are very specific reasons for both of those temperatures. Here's the logic. It's a tale of 2 temperatures, really. Scientists need results quickly, so they use 37 degrees, that's human body temp, which is the perfect temperature for these bacteria to multiply. But in a school lab, safety is priority number 1. Incubating at a lower 25 degrees dramatically reduces the risk of accidentally growing something that's really harmful to people. Okay, back to the Petri dish. 2 questions here. First, see that disc in the middle with no clear zone around it? What's its job? And second, based on these results, why would a doctor probably prescribe antibiotics A or B for a patient? And the answers here have to be really precise. That blank disc, that's your control. Its entire purpose is to prove that it's the antibiotic doing the work, not the paper itself. As for A and B, they're the best choice because they have the largest zones of inhibition, the biggest clear areas, which means they were the most effective at killing the bacteria. Simple as that. Now for the last 2 questions in this section, and they require a bit more thinking. First, how could you turn this visual result into actual numbers, make it quantitative? And second, here's a classic scenario. A year goes by, and suddenly antibiotic B doesn't work anymore. What's the biological reason for that? For the first part, to get numbers, you just need a ruler. You simply measure the clear area. The mark scheme says measuring the diameter or the radius is perfect. For the second part, the one keyword the examiner is looking for is resistance. The population of bacteria has evolved resistance to that specific antibiotic. Okay, we're on the home stretch now, the final part of question 3. We've talked about antibiotics working, but how do they actually do their job? This question wants the biological mechanism. Now this is a 3-marker, and the question itself gives you a massive clue. It tells you the antibiotic damages the cell wall, which makes the cell burst. Your job is to explain the steps in between. Why does a damaged wall lead to a burst cell? You've got to connect the dots. And here's how you nail all 3 marks. Point 1, you state that water rushes into the cell. Point 2, you've got to name the process. It's osmosis. And point 3 is the clincher. You explain that because the cell wall is weakened, it can't handle the pressure from all that incoming water, so, pop, it bursts. You need that full sequence to get the full 3 marks. So after taking that entire question apart, what have we actually learned about exam technique? It really all comes down to knowing exactly what the person marking your paper wants to see. Just remember the key lessons, right? For 1-markers, be sharp and to the point. For the longer questions, tell a logical story. And for anything about practicals, you've got to know your safety and your controls. The mark scheme isn't some big secret. It's a recipe. If you follow it, you'll build the perfect answer every time. So think about what we just covered. Which of those processes do you feel most confident explaining now?